I study proteins, which are these amazing molecular machines. Most proteins don't act on their own. They need partners. And what the protein structure does is it determines which partners and which other proteins it interacts with, which eventually leads to certain responses. These protein-protein interactions also play a key role in different diseases, such as cancer, neurodegeneration, and even the way that pathogens interact with us. Most drug discovery programs aren't really designed or haven't been planned to handle protein interactions because these are really large molecular surfaces. And so my group and many others are examining a complementary approach to what exists currently in drug discovery, which is to use the proteins themselves to inspire new molecules and new drugs. So what my group contributes to and what we work on is the development of new chemistry to stabilize particular molecular structures so that we can block specific protein interactions. We work right now on something called the Nipah virus. It's part of a family of viruses that is particularly nasty right now in Australia and Southern and Southeast Asia. It's really high mortality rates. Like think things closer to Ebola in the sense where we're talking like 30 to 50% mortality rates for these outbreaks. And thankfully, there's enough surveillance that they seem to be contained, but at the same time, it's obviously a huge issue, and having a way to treat someone that's been infected is a big deal. I'll say first off, we're a chemistry lab, so we don't work with any live viruses, especially those. They would be biosafety level 4, way off the charts, nothing we could reasonably work with. But we know which human proteins they interact with. So the idea is we know what virus we want to target, we know what human proteins it interacts with, so we're going to mimic the human protein with this much smaller synthetic peptide, and it's going to block the ability of the virus to enter the cell, which is the first part of infection. So if it never enters the cell, it can't infect you. The drug development pipeline is long and complicated. If our molecules get there, that's fantastic. Otherwise, what they often will do is provide helpful tools, just fundamental tools that we continue to use around the research lab to understand some of the underlying biology, which then could fuel development of other types of molecules.